it's my great pleasure to introduce Mathieu Groussin, if I pronounce this correctly, from the MIT in Boston, as well as at University of Kiel, talking about the Global Microbiome Conservancy. Over to you, Mathieu.
Thank you. Thank you very much, Manuel, for the introduction. Can you all see my slides okay? All right. Great. So, yeah, thank you so very much to the organizers, Pascal and Karina, for inviting both Mathieu and I together to present today on the Global Microbiome Conservancy, which is an initiative that we co-founded together. So before starting my story, I'd like to make everybody aware that all I'm going to present today really is the result of a joint effort and of my collaboration with Mathilde, who's still a postdoc in Icons Lab at MIT for another few months. So our research focuses on the human gut microbiome, and over the last decade, the research community accumulated a lot of knowledge on the beneficial effects of the human microbiome on host physiology and how many chronic diseases such as IBD, diabetes, or liver disease are intimately linked and connected to perturbations in our microbial ecosystems or to specific microbial functions. But the immense majority of this knowledge is derived from the microbiomes of majority mostly Caucasian populations in the US and Europe. And this is actually quite reflected by the imbalance in representation of major ethnic groups in big microbiome projects such as the HMP, or also illustrated by the imbalanced geographic distribution of the number of ongoing clinical trials that today involve the microbiome around the world. And so this bias in representation raises obviously ethical concerns, but also greatly contributes to the propagation of healthcare inequities. Globally, the predictions for the evolution of the incidence of non-communicable disease associated with the microbiome is quite alarming because about 70% of the human population is predicted to live urban lifestyles by 2050. So many populations in low and middle income countries will rapidly shift in lifestyle and are then likely to face rapid increase in the incidence of both microbiome perturbations and non-communicable diseases associated with this new lifestyle. So from a global health perspective, it's quite urgent to better understand, this relationship between lifestyle, non-communicable disease, and host microbiome systems. So to tackle all these problems at once, we created the non-profit Global Microbiome Conservancy initiative at MIT, which I will name GMBC for the rest of this presentation. So about 6 years ago, Mathilde and I co-founded and built from scratch this international consortium with the goal of biobanking and understanding the global biodiversity of human gut microbiomes. So what does the GMBC do? We have 4 cornerstones. First, conservation of this microbial diversity that is disappearing worldwide due to industrialization, and I'll explain how we do that later. Obviously, representation of the diversity of human populations, including the ethical use of their microbial diversity. We're also involved in capacity building with partner institutions, and obviously, we're engaged into advancing knowledge through science and research. So far, Mathilde, myself, and collaborators in the GMBC consortium traveled to 34 different communities of various lifestyles worldwide, and recruited about 1,300 healthy individuals in these communities. So with stool and saliva samples, we collected a wide range of metadata and generated both human genetic and multiomic microbiome data, and built also a biobank of nearly 10,000 cultured bacterial isolates from the gut for long-term preservation, and also generated their associated whole genomes for these bacteria. So this is here on this slide, you can see a map of places where we already collected samples shown in yellow dots, and places where we have ongoing discussions and collaborations to plan future samplings shown in blue dots. And I couldn't resist really showing you a few pictures that our collaborators in Pakistan, Nepal, and Argentina, and Indonesia recently sent us about their ongoing trips to isolated areas to collect samples among very isolated communities. So on the top left here, you can see that we recruited individuals among both industrialized and non-industrialized countries, and within those, we recruited cohorts among urban and rural settings. So we recruited cohorts with a great diversity of lifestyles and diets, as shown in these two multivariate plots here. And so what do the data really on this global microbiome and human diversity tell us? So here on the right of this slide, I'm just giving you a snapshot of some of the analyses that are ongoing and that we're running. So you can see how much diversity in microbiome compositions our dataset captures, and the impact that industrialization or lifestyle has on these compositions, where you have like rural and indigenous populations here on the far left, farmers falling into the middle of this first axis, and then the more industrialized and urban individuals falling on the right here in terms of compositions. So our cohort also captures a great diversity of in-host genetics, as measured here through exome sequencing and genotyping, which we compared here to data of the 1000 Genome Project. We also gathered and generated data on host biomarkers and host microbe interactions. So I'm illustrating this here with data on differences in the amount of immunoglobulin A secreted by intestinal cells between industrialized and non-industrialized individuals, which is much higher among urban and industrialized people, but also differences among these two groups, between these two groups, in the amount of IgA, immunoglobulin A, targeting bacteria between these two groups as measured through IgA sequencing. And so as I said and presented earlier, with our samples, we also built an extensive biobank of individual bacteria in pure culture paired with whole genome sequences. And so to do this, we developed culturing techniques and media to grow and isolate in anaerobic conditions thousands of gut bacteria. So up until our work, most of the isolate collections of human gut bacteria were biased towards diversity found in industrialized populations. But now our collection represents individuals with, are coming from individuals with a diversity of countries, lifestyles, and subsistence strategies, including many from non-industrialized and rural areas. But the collection of bacteria are also very diverse in terms of bacterial taxonomy itself, as you can see here in this phylogenetic tree, and contains nearly 500 different species across multiple phyla. And so what's great is that with this biobank of the isolates that are biobanked and paired with their genomes, we can have direct access to a realm and a great amount of totally unexplored diversity that we can investigate further with genomic or experimental techniques. And so today I chose to present you a computational analysis that we recently published, leveraging our data set of whole genomes of bacterial isolates. And so in this study, we investigated the impact, and that we co-published with all our cohort authors in the consortium. We investigated the impact of host lifestyle and industrialization on gut bacterial genomes through horizontal gene transfers, which is the non-vertical transmission of genes or genetic material from one cell, from one bacterial cell to another. So horizontal gene transfers, or HGTs, are particularly frequent when bacteria experience threats or environmental perturbations in the ecosystem, because they can quickly acquire new functions by picking up genes from the outside in order to adapt to these perturbations. So what we did is constraining our analysis to the most recent gene transfers that occurred between now and just 2 to 4 generations ago. And we found an extensive amount of HGTs occurring within our own microbiome, confirming that the gut is really a hotbed for gene exchange between our gut bacteria. But we also found, surprisingly, that bacteria exchange genes much more frequently when they occur in more industrialized and more urban cohorts. The functions that they exchange are also quite different, reflecting very well the level of industrialization of the host. And so overall, this analysis really suggested that gut bacteria continuously acquire new functionality during our lifestyle, including genes involved in antibiotic resistance, for instance, or carbohydrate degradation. And that the high rates of gene transfers may be a recent development in human history linked to industrialization, potentially associated with increased perturbations in the gut ecosystem. And so now I would like to take a couple of minutes to discuss with you about the future of the GMBC, because as an academic initiative, we felt that we reached at this level a milestone in our mission, and we now want to scale up the scope and impact of the GMBC on microbiome science and participating populations. And so in just a few weeks, we will announce that the GMBC is spinning out of MIT to be stood up as a program at OpenBiome. So OpenBiome is a nonprofit organization and the first public stool bank dedicated to provide safe access to fecal material transplants for patients with recurrent Clostridium difficile infections, and also to catalyze basic and clinical microbiome research. And I think that since it started in 2013, it has manufactured and provided more than 60,000 FMT preparations to treat Clostridium difficile patients, but also supported more than 30 clinical trials. So by building the GMBC program at OpenBiome, we aspire really to ensure that everyone everywhere is benefiting from microbiome science and research. So the mission of the GMBC at OpenBiome will be to diversify and accelerate global microbiome science and build new paradigms for ethical research by working with communities, collaborating researchers and experts in bioethics. So OpenBiome is really strongly supporting the program by committing a dedicated team for field collection, microbial biobanking and phenotyping, and dissemination of bacterial strains to the research community. So for this, the GMBC program will be using OpenBiome's expertise and know-how in biobanking and distribution of biological materials, and will be positioned to start providing access to this resource later this year to advance research. So importantly, we have built a science and ethics advisory board, which will be composed of scientific and ethics thought leaders to guide the program, and I will be very honored to chair this board. So the first priority of this board will be to develop a benefit sharing model that will be funded by the proceeds from the use of the library to ensure participating communities benefit from the distribution and use of the diversity that we'll distribute to third parties. So we're extremely excited to announce this expansion of the GMBC and to see how it progressed since its inception. And so stay tuned really for more info very soon. So for now, I'd like to thank, of course, Mathilde, but also several members of the ERICOM lab, as well as Ramnik Xavier, the organizers to this symposium as well. We're also very thankful to all our collaborators and participants of the GMBC, all folks at OpenBiome who are building the future program, and to all our sponsors. And as presented by Manuel earlier, we're going to start our professorship positions this February 2022 at Kiel University in Germany. So we have open positions for PhDs, postdocs, and technicians. So if any of you in the audience are interested in our work and want to join our lab, feel free to reach out. Or if you are interested in joining the GMBC program and consortium, feel free to reach out to us as well. Thank you very much for your attention.
Thanks a lot, Mathieu, for this exciting talk. If there's questions, just a reminder, please ask them in the question and answer section. You can also do so in French or in Portuguese, so that will be translated. So we will have time for maybe one extremely quick question if there's something urgent upcoming.
Yeah, I have a question for Mathieu, if I may.
Hi,
Mathieu, it's a great talk and great initiative. And my question is, so are you biobanking the isolates, or you also keep a specimen collection?
Yeah, we actually biobank both. We have this tool in glycerol solution that we biobank, and the isolates themselves that we also biobank.
So we hope in the future you think of depositing your backup safety collection in the microbiota vault, where we also plan to do metagenomics of the samples and make the data available. So
right.
That would be, it's a great resource for a backup security collection.
So we
Thank
do.
you.
Yeah, thanks, Maria. Yeah, we do also generate all the metagenomic data from our samples. So you're talking about biobanking the original stool samples. That's what you're interested in here.
Both original stools and isolates, but
yeah.
Original stools we think are very important because we may not have the capacity to culture everything right now.
Right, that's correct. Yeah. So that's actually what we see as well. So we're developing techniques to capture the ones that are particularly difficult to get. But there's certainly room for like projects in such collaborations and such collaborations in the future. So we should definitely reach out and discuss about how we can make that happening.
Thank you.
Thanks
you. Great initiative.
And there's another important question in the question and answer section regarding also like the benefit sharing with the countries. But if Mathieu is joining the session for question and answer it in the evening, I would suggest for the sake of time to answer it there or in live. Thank you.